application of iron oxide in removing metal nanopollutants from contaminated industrial effluents. Okay, yeah. So, in this lecture, I'm going to compile everything that what we have learned. There are two kinds of magnetic nanoparticle we synthesized. The one is the water soluble, other one is the surface active. So we will try to see that how the surface active magnetic nanoparticles, apart from the biological applications, they can be used for certain applications, other applications, environmental related applications. Likewise, how the water soluble magnetic nanoparticles can be used. So what we learned, this is the summarization of hydrothermal synthesis. By using the hydrothermal synthesis, you can synthesize both kind of magnetic nanoparticles, water soluble or the surface active. The only difference is the stabilizing agent or the colloidal stabilizer. The colloidal stabilizer is going to determine if the magnetic nanoparticles are surface active or water soluble. When you are using typically Gemini surfactant, many of the Gemini surfactants, so they lead to the surface active magnetic nanoparticle synthesis, whereas any other amphiphilic molecule which you are using, provided that should be an amphiphilic molecule that has the ability to absorb on the surface of the magnetic nanoparticles, that will lead to the water soluble magnetic nanoparticles. Now we know the characteristic properties, we know the crystal structure, we know the unit cell, everything we know. And now we can see that under what circumstances we can use them for different kind of applications. So predominantly, suppose if you ask me, though I have shown you several images of the magnetic nanoparticle, predominantly this kind of synthesis where I have specifically mentioned in the lab experiment, Dr. Rajiv specifically mentioned that what should be the concentration ratio of ferrous sulfate, ferric sulfate, and that of the ammonium hydroxide along with the stabilizing agent. If you are choosing to keep that ratio close to that ratio, more or less you will be having this kind of nanoparticles. So shape and structure, I would say size particularly, the size will remain this. But if you are going to vary that ratio, you may get different size and different shapes. Apart from that, the shape and size also depend on the concentration of the colloidal molecule, that is the surface amphiphilic molecules. So if you are going to vary the concentration of the amphiphilic molecule, now you have learned that in your synthesis in the lab experiment, you use 250 mg of a Gemini surfactant in order to synthesize the surface active magnetic nanoparticle. So because we have standardized the condition accordingly to get the smallest possible size. This is the smallest possible size we are using. Smaller the size, higher is the surface area, and better is the applicability. That is the main purpose or objective for us to produce the nanoparticle with the smaller sizes. And that is why most of the samples, they lead to this much size almost in all the cases. Uh, as I mentioned you in the previous lecture, when we were extracting the blood cells, so I mentioned at that time that we prepared 8 samples of amino acid stabilized magnetic nanoparticles, and then we had approximately 4 to 5 samples of sugar derivatized magnetic nanoparticle. So they are the functional magnetic nanoparticles. When you are going to, if somebody asks you, what is the meaning of the functional magnetic nanoparticle? So functionalization is to put certain kind of functional group on the surface of the magnetic nanoparticle. That could be amino acid, that could be a colloidal amphiphilic molecule, or that could be any polymer. So that is the functionalization. I mean to say that If you do not functionalize this, so then in fact you have the bare surface of the nanoparticle, and the bare surface of the nanoparticle may not be capable of carrying out a precise applicability for a particular kind of application. So therefore, if you want to have uh, 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 if you want to use magnetic nanoparticle for a specific kind of application, then obviously the magnetic nanoparticle has to be functionalized with a specific kind of functional group that would help to facilitate the application. Now, uh, you might be remembering that we use the polyethylene oxide, polypropylene oxide, and polyethylene oxide block copolymers for the templating effect, as I mentioned in the first lecture today. So these polymers, they have the hydroxyl group. When they are going to adsorb on the surface of the polymer is going to adsorb on the surface of the on the magnetic nanoparticle, then these functional groups are available. So now you can use these functional groups for performing certain kind of applicability. This is the meaning of the functionalization. So I'll show you that how we can use such kind of a molecule for environmental applications, and among them the extraction of industrial dyes. You know, you are using dyes everywhere, so you need to change the color of your clothes, so you are taking them to a person who is dyeing it, changing the color, and then everything goes to the drain because dyes are water soluble, most of them, and because they are the organic molecules, so therefore they are the toxic molecules, and our fresh water resources are going to pollute. Dyes are highly toxic and polluted. Dyes are highly toxic and one of the most pollutant of our natural resources on the planet. It is not limited to this country, but everywhere you go, dyes are highly pollutants, and therefore, if you search the publication related to the cleaning of the dyes or cleaning of the contaminated water with dyes, you will see series of publications. So many papers have been published for the cleaning of contaminated water with dye molecules. You won't be able to imagine. So everybody, so means this is a very hot area, and what we have done, we have devised certain method on the basis of the magnetic nanoparticle to clean the contaminated water that is contaminated with a dye molecule. So I'm going to show you that one, and that is shown here in this flow diagram. So these are different samples of the dye solution. This one is highly dark purple dye, blue one, and you may say that this is close to the pink dye. And this is, you may assume that all these solutions are the contaminated water. We want to clean them, right? How we are going to clean them? We have two methodologies. I'm going to show you two methodologies, and by using the magnetic nanoparticle, and how can we clean them? Here we have used water-soluble magnetic nanoparticle, polymer functionalized magnetic nanoparticle. What we do, we synthesize these magnetic nanoparticle exactly the procedure which you have done in your lab experiment. So then you got this kind of solution when you completed the experiment, and applying with the magnet, you extract the magnetic nanoparticle and you purify it. Essentially, you have purified magnetic nanoparticle. What you can do, you take the dye solution that is contaminated water, you put it into here. Here you have the magnetic nanoparticle. You keep them as you could see over here. So I have taken this one. You may say that suppose we are picking this solution, we transfer this solution into the container which contains the pure magnetic nanoparticle. Functionalize this. You have to keep in your mind that These magnetic nanoparticles are functionalized with OH groups because of the block polymer that is adsorbed on the surface of the magnetic nanoparticle. What happens then, if you keep it some time, eventually you see the dye color is going to be faded away. As more and more dye is going to be absorbed by the magnetic nanoparticle, the dye color is going to vanish. In some cases, entirely the whole dye is completely absorbed on the magnetic nanoparticle, leaving behind a completely transparent solution. In some cases, you may not have a complete extraction and therefore you may have light color left. It means that some amount of the dye is there in contaminated water. So anything can happen. I mean to say that if you really want to clean the entire solution, obviously you need to have the higher amount of the magnetic nanoparticle. If you take the greater concentration, obviously eventually a point will come when you can remove the entire dye molecules and the solution is colorless. What you can do thereafter, you need to quantify that this much amount of the sample of magnetic nanoparticle is capable of extracting how much amount of the dye molecules. This is the quantification. If you have to tell somebody that I have a new methodology to clean the contaminated dye molecule, then a person will ask you, okay, you tell me how many grams you are taking the magnetic nanoparticle, putting into the dye solution, and how many grams of the dye has been extracted. That is the meaning of quantification. So, once I have this dye-loaded magnetic nanoparticles, I can quantify with the different elemental analysis, transmission electron microscopy, scanning electron microscopy, it is XPS, X-ray photon electron spectroscopy. So all these studies can help me to see where the dye molecules have been adsorbed on the surface. They are associated with the hydroxyl group or on the surface. In what form? What is the conformation or what is the molecular status? Is the dye molecule remains intact or it is decomposed? And all those things I can analyze by going through these analysis. Apart from that, I can calculate how much amount of the dye has been extracted by taking how much amount of the magnetic nanoparticle. And suppose you may ask me, how long you have to keep it in the dye solution? Uh, we have performed the experiment and we have seen that some of the dyes are extracted within just few minutes and the solution you are going to get is transparent, but some of the dyes take a very prolonged time. It means that overnight or maybe 3 to 4 days eventually when you get the clear solution. So it means that the extraction of the dye depends on the dye molecule, structure configuration of the molecule, and how it is extracted by the functionalized magnetic nanoparticle. So therefore, it is time consuming or a rapid reaction. It altogether depends on the molecular interactions between the functional groups and the dye molecule. If the molecular interactions are stronger, as I mentioned in my previous lectures, so then the extraction is instantaneous and rapid. But if the molecular interactions are weaker, then it's a time consuming. So dye molecules are taking time in order to upload on the magnetic nanoparticle. So this is the first method. We call it static method. It means that we have added the magnetic nanoparticle and we observe only. It's a very simple experiment and we observe that the extraction is happening. There is a second experiment over here. This is very important. The second experiment is the titration experiment. So exactly the same kind of titration which you perform. So in this experiment, what we do, we take the magnetic nanoparticle suspension, purified, we take the purified magnetic nanoparticle and we add pure water and we have the only pure magnetic nanoparticle suspension. We take a burette and in the burette we add the dye solution. right? So we keep adding the dye solution. After each addition, we place the magnet and extract the collect the nanoparticle and measure UV. After another addition, we, we we perform the same thing. It means that what we are doing. So we have a burette and we have a beaker. In the beaker, we have magnetic nanoparticle. So what we are doing, we add 1 ml of dye, we shake it, and then we apply the magnet. All the magnetic nanoparticle are taken aside by the magnet, and then we pull the solution and we measure the UV. Again, we put back the solution. Again, we add 1 ml, that is 1 ml of the dye solution. We perform the same thing. Eventually, a point comes. Eventually, a point comes. Yeah. When we keep on adding, when we add the first 1 ml, suppose we add 1 ml of this solution is completely transparent because the smaller concentration, when we start adding the smaller concentration, the first lot of dye, when it goes into the solution, it is instantaneously absorbed the magnetic nanoparticle. When you collect it, your solution is completely colorless. If your solution is completely colorless, then you won't be having the absorbance of the dye molecules, right? Eventually, you keep on doing it. A point comes at a certain addition, you see the color appearing. The color of the dye starts appearing. This is the end point of the titration, right? So it demonstrates that at this concentration, you cannot you cannot add any more amount of the dye. So the magnetic nanoparticle concentration, they are already saturated with the dye. So they have extracted whatsoever the maximum dye they could extract. Now we need to find out this concentration. What is the meaning of this concentration? Quantitatively, quantitatively at the end point, suppose I say that I have taken 1 gram of magnetic nanoparticle, 1 gram of MNPs, magnetic nanoparticles. At this point, uh, just a minute, sorry about it, not 1 gram. I should put the concentration units. So I should say that, okay, I have taken 40 millimolar the magnetic nanoparticle. So this is the lot where the total concentration is 40 millimolar, right? And how much dye has been extracted? Because I am adding dye and this is the total volume, I can calculate the final concentration of the dye. I may say that, okay, this is probably 2 millimolar of dye, right? What I can do now, I can convert into 40 moles of MNPs, particle. Likewise, I can convert the millimolar into moles. And I can convert, because if the volume is constant, let me say 2 moles, okay? And 2 moles of dye. Now, once I have the moles, I can convert into grams. So it is the grams of magnetic nanoparticles divided by grams of dye. This ratio is required for any kind of industrial application. The person who is running the industry, he or he may not be chemist. He only needs to know that how many grams of magnetic nanoparticle I have to add in a dye solution and how many grams of the dye molecules are going to be extracted. So that is why this ratio is required. And you could see that with the help of this simple titration procedure, you can this very important ratio. Now, you can have this ratio for all the dyes, right? Or if you want to choose some other kind of functionalized magnetic nanoparticle to facilitate the extraction, You can check. Whatsoever is giving you the best ratio. Best ratio. What is the best ratio? The minimum amount of the magnetic nanoparticle consumed to extract maximum amount of the dye, that is the best ratio. Am I correct? Because you need a magnetic nanoparticle sample that is much more efficient to extract maximum amount of dye. That is the most appropriate sample. So that is our purpose to check that. So here is the experiment which I was mentioning. As I keep on, as I start the addition, initially there is no absorbance. Eventually you see the dark yellow color. The dark yellow color, yeah, it is this one. Though there is one more over here, but this addition represent this much concentration. After this, there is a bright green, and eventually this is. It means that end point has come over here. It is a 0.08 millimolar of the brilliant blue dye. So this is brilliant blue dye solution. So this is the concentration. The end point of this titration is somewhere over here. So now you could see that what we have done, we have titrated, we have performed the titrations under different pH conditions. The neutral pH, basic pH, and the acidic pH, just to see that which pH environment is going to give us the maximum extraction. What we have seen over here, you see that. So in the case of the acidic pH, yeah. So you see that in the case of acidic pH, close to 1 millimolar of the dye is being extracted. It means that's very high in comparison to the neutral and the basic. Neutral and basic, you may assume close to 0.1 millimolar, 10 times high. If you are performing this titration under acidic condition, so you can extract dye up to 10 times in comparison to the neutral medium, right? And so we took 6 dyes. It's 1, 2, 3, 4, 5, 6, and we performed the titrations just to find out the which dye is giving the best results. So we found that this is the brilliant blue that is giving the best results. So the extraction is close to 1 millimolar for this one under pH, acidic pH condition. Likewise, this is, I forget its name, what is, excuse me? Yeah, black tea, sorry about that. Actually, it just went out of my mind. So it is Iroquois black tea, right? And the black tea also performing excellent results. So though it is not equivalent to that of the brilliant blue, but still close to 0.8 millimolar. So in comparison to the required sample of the magnetic nanoparticles, you can extract it. Then we have methyl violet. Methyl violet is not working appropriately. It is working more in the basic condition rather than the acidic condition. You see, the acidic condition is the minimum extraction. Whatsoever we extraction observed, so that is in the basic condition. And surprisingly, all these 3 dyes, this is methyl orange, uh, rhodamine blue, and this one is bromophenol blue, correct. So all these dyes were not extracted at all under any pH condition, acidic, basic, or neutral. So what is the meaning of the functionalized nanoparticles application? I say that you cannot expect that the functionalized nanoparticle, magnetic nanoparticle which you have synthesized, is going to be is going to work for all kinds of dye. That is not the case, right? It is the functional group which will decide that how it interacts with the functional groups of the dye. Wherever the functional group that is on the magnetic nanoparticle, if it has the stronger interaction, in this case it has a stronger interaction with the brilliant blue and this dye, so because of that the extraction is maximum. But This functional group has minimum interaction, so extraction is not happening with the titration. But if you perform the static extraction, like the static method, so then all dyes are extracted, though they are not equivalent. But even those not shown in the titration method, so they are being extracted. The reason is the titration method is very rapid. You are adding dye, maximum 3 to 4 minutes you are going to wait, and you are going to measure the UV. But in the static method, you will wait for overnight, more than 12 hours or 24 hours. That is not the the uh, 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 the time period you are going to consume for the titration. So titration is a rapid method, and if you want to sell this technology methodology to some industry will prefer the titration method rather than the static method. So they will prefer that we need a kind of a rapid method which can instantaneously extract the dye in a shorter period of time. So, so bottom line, this is the way we choose, or this is the way the functionalized magnetic nanoparticle work. The main purpose to show this is to just tell you that how we can design synthesized functionalized magnetic nanoparticle for a particular application, and on the basis of that application we could see that how much applicability this kind of functional magnetic nanoparticle can have, and in this way we can further modify or improve the method. So how the extraction is happening? The brilliant blue is showing the maximum extraction in the acidic medium rather than the basic medium. Here is the story. In the basic medium, actually, the hydroxyl groups are deprotonated, right? They are negatively charged. The negatively charged group, they are not going to interact with the sulfonate group. So this is the sulfonate group of the brilliant blue. So therefore, they are going to interact with the positively charged nitrogen here. But the problem is this positively charged nitrogen is screened with a bulky group that is screening the electrostatic interaction. Remember I told you, it's not always the electrostatic interactions is going to work. You have to find out that if there is any bulkiness, screening, or the steric hindrances which are impeding the electrostatic interaction. If that is the case, you may not have a sufficient kind of extraction. On the contrary, you see over here, when you have protonated hydroxyl group in the acidic medium, this is not a problem. So for the protonated hydroxyl groups, so the electrostatic interactions can easily be performed with the terminal group like this sulfonate group, and hence the extraction is very rapid. So I mean to say that these, you need to have the knowledge of, the understanding of every kind of fundamental understanding which is involved in the procedure to understand or to apply it appropriately. So I mean to say that the knowledge of the basic chemistry is very, very important. If you are clear about the basic concept, I don't think there is any issue when you have to perform any kind of research activity. So this is the one, this is the example of the extraction of dye. I just wanted to show you that how functionalized magnetic nanoparticles work while performing certain kind of applications. And this is specifically being used by using the block polymer or block polymer. So these are the water soluble magnetic nanoparticle. Sorry, yeah. Another thing, suppose, so you might be asking me, what is the purpose of transmission electron studies or scanning electron microscope? You see the images. Suppose if you ask me, where are the dye molecules? We can see them. These are the magnetic nanoparticle now carrying the dye molecules. You cannot see them in the black and white. But if you apply the elemental mapping, these colored images are known as elemental mapping. Elemental mapping means if I apply to see only the iron, then I will see the color of the iron. I'll see a colored area. Iron is present in the sample. So this color, you can have any color. I mean to say you can change the color in the instrument, but if you see the colored area, other areas are completely dark. It means that that area belongs to iron, right? So you see that these are the iron oxide nanoparticles. So therefore, iron is there. Oxygen is also there. So it is iron oxide. Now you want to see the carbon. The thing is that you need to know where the carbon is, okay? The carbon is here, methylene group, methyl group, methylene group, they are all carbon. It means the carbon is in the polymer backbone. Apart from that, the carbon is in the dye molecules. So your dye molecules, they have all carbon. It means that this carbon is because of the carbon from the polymer as well as the dye. You cannot differentiate that which carbon is of the dye and which carbon is the polymer. But at least you see the ring structure. Can you see the ring structure? The ring structure mesh shows that the carbon is coated around the nanoparticle. That is why you have a ring structure. Because if it's a roughly polyhedral nanoparticle, it means that if it's coated with the polymer, carbon is originating from there. And if the dye is being absorbed, dye is going to interact with the polymer molecule, so therefore the carbon is there. So since inside the ring structure, the dark areas, because it is more iron oxide, so therefore the emission of the carbon is not that much, so therefore that is not a bright green area, though the carbon is having a layer around the magnetic nanoparticle. What is unique only to the dye? That is nitrogen. Nitrogen is only available in the dye molecules. It's neither the part of iron oxide nor the part of nor the part of polymer. Polymer is not having nitrogen. No nitrogen functional group in the polymer molecule. And there is no nitrogen in the magnetic nanoparticle, it's iron oxide. So therefore, nitrogen is here, here, and here. So therefore, the nitrogen which you see is the part of the dye molecule. So therefore, in the sample, you see the mapping of the nitrogen, that is only because of the dye molecule. That shows, now you see that shows the extraction of the dye. The nitrogen mapping clearly indicates that it is available on the surface of the dye. You see the circle area, you see some of the tiny circle area. So therefore, it is the part of the shell that is created around the magnetic nanoparticle. And apart from that, you have the sulfur as well. Though the amount of sulfur is very less, this is the only functional group, sulfonate, that is the origin of the sulfur mapping. And so therefore, both the nitrogen and the sulfur mapping confirms the presence of the dye. So I mean to say, by applying all results, you come to the mechanism of the extraction. How do we know that the nitrogen or the dye is not decomposed, right? If it's not fragmentized, I'll say it looks that the dye molecule is intact. There is no decomposition because we have the nitrogen emission as well as that of the sulfur. So we could map the sulfur as well as the nitrogen, and we can also quantitatively on the elemental basis, we can see how much is the atomic percentage of nitrogen, sulfur, and carbon, and we can see that, and we can calculate the same ratio. The ratio we have calculated over here, the same ratio we can calculate over there from the imaging study. So this is the advantage of imaging study, but you need very high resolution to get these images, and very highly sophisticated transmission electron microscope. Usually they are not available because they are very, very expensive. So you have to perform the experiment very carefully under uh, entirely non-contaminated, because even a single dust particle would create the elemental emission from the element, which you will be surprised that why this elemental emission is there, and then you will realize that it is because of the contamination. So I mean to say that when you have to work in a very clean environment, and no matter you are working in a nanomaterial, any kind of research, inorganic, organic, or physical chemistry, so you have to have a clean environment and non-contaminated environment to quantify the research. So sustainability, and remember I told you that sustainability in the first few lectures is a very important aspect now. Now we want that we should have whatsoever kind of action we perform in our day-to-day life, we should have certain degree of sustainability. I mean to say simple action is when you are using the white sheets. So we want that the white sheet after the usage, we don't throw in the garbage. They could be recycled to produce the fresh white sheets. The plastic we are using, it should be recycled, and we should have another lot of the fresh plastic which can be used for another purpose. So that is the meaning of sustainability. But it's the same way. So if we could reuse, now you know that when you have to use the reverse osmosis for water purification instrument, you have everybody in your home, right? So you are changing the cylinders. So I don't know if they are reusable or they are regenerating them, how they are doing that. But in this case, you can easily do. How? See over here. So here is the brilliant blue solution at pH 1.5. When I'm going to add magnetic nanoparticle, in pH 1.5 extraction is highly facilitated. Instantaneously, the whole extraction is going to take place and your solution is completely transparent. Okay, and you could see it. Here is the actual solution. And when I apply the magnet after adding the magnetic nanoparticles are over here. You see the transparent solution? Entirely the solution is transparent, right? What I do, I pick the nanoparticle. It means nanoparticle have picked all the dye molecules from this solution. And I pick the nanoparticle, put it in the basic solution. The pH is 12.5. I know already from my results the the interaction of the dye and the magnetic nanoparticle least in the basic region. So therefore the dye will be released. The entire dye which was extracted over here is further released into the same bottle because you have changed the pH. What you could do, you again collect the magnetic nanoparticle purified. I mean to say that this is the purified water. You have thrown away the dye in some, I would say, the discarded bottle, right? And then again pick the same lot of magnetic nanoparticle, put it in the fresh lot of contaminated water. You can recycle the same lot of magnetic nanoparticles. You don't need any fresh magnetic nanoparticle. And here is the cycle. So we have performed up to 4 cycles, and the concentration of the released dye, that remains more or less same, close to 1.5 millimolar in all the cases, and this is the UV. So for all the 4 cycles, the UV plots are quite close to each other. So I mean to say, this is known as sustainability, right? So what I want to say that in our research, we should now be more careful that how I can have, how I can include the sustainability at some point in my research. By adding the sustainability, it means that you are generating a new field of the research so that your natural resources, our natural resources, are not going to be exhausted. That is the main purpose of the sustainability. Oh, sorry about that. Uh, I wanted to show you some other application by using the cyclodextrin over model pollutant. Earlier I showed you the extraction of the model pollutant by using some kind of surfactant, but this was by using the cyclodextrin. But I have to stop over here because the first application, I never realized that it's going to take so long. Anyway, so I should I should I should keep it off, right? And I should finish it over here. So hopefully you feel this is a very interesting application where you can have the sustainability as well. And now I'm directly going to the conclusion part. Surface active magnetic nanoparticles are a new class of nanomaterial with interfacial application. You have already seen the interfacial application. The most important among them was the extraction of the blood cell. That was directly related to the bioapplicability. They remove pollutants without incorporating in aqueous bulk. This is what we saw when we took the gold and silver nanoparticle as model pollutant, and we try to use the surface active in order to extract the metallic pollutant, and those metallic pollutants means the industrial effluents from the metal industry. So how we can clean that contaminated water which is with that of the industrial effluent. They possess enormous potential to use in the biological application at biointerfaces. So I have shown you just one application, that is the extraction of blood cell or the protein film formation, but there is huge range of bioapplicability at the biointerfaces. Among them, usually the magnetic hyperthermia, MRI that you are being means it is a very common. So all these applications particularly require some in-depth analysis of in-depth knowledge of biology. So this is all about the entire course. Hopefully you have understood some part of the course, if not the entire course. And what I believe that should be useful for you some point of at some point in your career, whether you are pursuing research or not. That is not important. But even having the knowledge of material science or chemistry, that is very, very important. So at the end, what I would like to suggest you from my experience, that how I became a scientist. I feel still not a good scientist, but still I'm trying at this age. It's a journey, long journey of more than 30 years. I still remember when I was student doing PhD in Punjab University. So it is more than 30 years ago. So at that time, I never thought that I'll be able to publish this much and I could do this much research. But how it happened, and if you ask me just in one sentence that how it happened, I'll tell you that only the basic chemistry. If you understand the basic chemistry, fundamental chemistry, first year chemistry correctly, quantitatively, there is no problem in the research field that you cannot solve. This is, I'm telling you, from my experience. And the day I started understanding the fundamental chemistry, it, it, it was not the case when I was doing my PhD. I, I had so much confusion. The basic chemistry was not clear to me. The fundamental chemistry at that time was not clear to me. But eventually when I started teaching and slowly reading textbook and everything. So the most advantaged, I would say the most important time obviously was when I was in North America. We have excellent textbook for the introductory chemistry courses, and with clarity of understanding the concept, applying quantitatively, right? So that helped me a lot. So it is not that easy to buy those textbooks because they are very expensive. But I would suggest you that you can tell your library or to your department at least one or two copies of such kind of recent textbooks which are being used in good universities in North America or in United States. So they are not that expensive if you have to buy only two copies to keep in the library. So one copy costs usually our 10 textbook costs, which are required by the every student. Every student has to buy it. It's $150 close to, and sometimes they reach to $200 per copy. So there it is required, and every student is required to have that before they enter into the course or come to the first class.
So
So
here we
here we
cannot
cannot impose such expensive textbook, but I would say that that is very important thing. And eventually, though textbook really help, but if you are not, if you do not have an interest in chemistry, then textbook cannot help you, right? So therefore, you have to create your interest in chemistry, the fundamental chemistry, to understand it,
and
and
eventually
eventually try to apply it. So at the end of the course, so I'm so grateful for you, all of you, who spared your valuable time to go through this course and try to understand the aspects which we have included, and hopefully they will be useful for you in your future career. Thank you so much. Any question, please, if you have.